Hello everyone. Have you ever wondered how transactions on safe platforms take place on secure basis? If not, let me tell you. Our project is an application of this. The topic we have chosen is DNA Steganography. The members of the group who have worked on this project are Abhishek Abhi, Aditya Iyer, Anjali Chandwani, Kunj Bua, Mahi Shah and Sakshi Shah. Now let's begin. What do you think is the need for encryption? Encryption is crucial for protecting sensitive information, maintaining privacy, and ensuring data security. Encryption is essential for data security, privacy protection, and confidentiality. It plays a crucial role in today's digital landscape. Steganography is method of encryption. What do you think why steganography is important? Steganography adds an additional layer of security by making the hidden information less apparent. Unlike encryption, which indicates the presence of encrypted data, steganography can make it difficult for adversaries to even detect the existence of hidden information. Where do you think this technique originated from? In 1994, Leonard Edelman first proposed the idea of DNA steganography. DNA consists of four bases AGCT, adenine, guanine, cytosine, and thymine. In our case, we have chosen the binary values of AGCT as A is equal to 00, G as 10, C as 01, and T as 11. Now, let me explain you the process of DNA encoding. Suppose the input string is M. Divide M into characters M, which is equal to M1, M2, M3, so on till Mn. Convert each character to its 8-bit binary counterpart using the ASCII values. Create K1 by randomly selecting a value between 0 and 255, and then the key is transformed into 8-bit binary sequence. K1 and each key in M is XORed. A sample DNA sequence S is selected randomly and converted into a binary bit sequence. Generate a random number, preferably a small value K2. Here in our case, we have fixed value of K2 as equal to 2. And then divide the DNA sequence S into segments with segment length equal to K2. Insert the first binary value of A at the start of the first DNA binary segment, followed by second binary value of K1 and so on. At last, we concatenate all the values of binary sequence and then use the above table to generate a bogus DNA sequence. Now let's move on to data decoding. Using the above table, we converted the incoming fake DNA sequence into a binary sequence. Then further, we subdivided the binary DNA sequence into segments with size equal to K2 plus 1, here in our case, which is fixed to 3. Now we concatenate the first bit from each segment to obtain significant bit B. Then we XOR the first 8 binary bits of B with K1, the original key, and then convert the DNA sequence binary bits to ASCII text values. There are many advantages of DNA steganography. It's used in large-scale data storage. It can be also used over a long period of time while being intact because DNA molecules are very stable and can endure for thousands of years when preserved under appropriate conditions. There are many real-life applications of DNA steganography. It can be used in the field of digital forensic to conceal data and preserve evidences. It can also be used for secure communication and information exchange. It is also used in scientific research, historical documents, or storing personal data. What do you think how DNA steganography can be commercialized? It is essential to create dependable and efficient techniques for finding and releasing the locked information in DNA sequence for commercial applications. Strong encryption techniques and security procedures are necessary to ensure the integrity and confidentiality of the concealed information. The use of digital image steganography has become more widespread nowadays. Since the human eye typically cannot detect minute changes to the least significant bits of the picture pixels, many strategies were developed to incorporate data into these bits. Using this technique, it was feasible to hide secret messages within images without significantly altering their appearance. Thank you.